Hello, everyone, and welcome to today's broadcast, Using CRISPR and Stem Cells to Treat Genetic Disease, presented by Bruce Conklin, Senior Investigator, Gladstone Institute of Cardiovascular Disease, Professor, Department of Medicine, Division of Genomic Medicine, University of California, San Francisco. I'm Alexis Krause of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Conklin. I will now turn the presentation over to him.
Hello, this is Bruce Conklin, and it's a pleasure to be here. I'm going to be talking about using CRISPR in stem cells to treat genetic disease. I'd like to just start with a conflict of interest statement. I'm involved in several companies, as indicated here, and then nonprofit organizations. I also would like to start with my list of the contributors, people who've contributed to this project, or in particular the ones in yellow, both people in my laboratory as well as recent alumni, and then importantly the collaborators who provide us with the critical tools and advice along these projects. I think my lab actually works between combining two disruptive technologies, and remarkably, they're just really bookmarked by two different papers, as indicated here. One in 2006, 12 years ago, and then in 2012, the invention of CRISPR. And it's these people and the technologies which are illustrated here, where we work, in the sense that we take iPS cells and we use them as models of human disease, and then we use CRISPR to test hypotheses about disease within iPS cells, and then more recently using CRISPR to see whether or not we can use CRISPR as a tool for actually curing some of these diseases. So it's between these two technologies and the intersection that we work the most. So why use iPS cells? And these are the stem cells that we work on, primarily because they can make many different types of tissue that were just inaccessible before. My lab in particular works with making neurons that are, for instance, motor neurons, as well as I'll tell you about our project with retinal pigment epithelia. And we've also worked with cardiac muscle. These cells and the other ones listed here were just inaccessible from defined humans with disease. And it's these sorts of cells which really provide a dramatic setting for us now to proceed and ask questions about disease. What the iPS cells themselves can not only be directly differentiated to one particular type of cell, but it's also using organoid cultures where cells are now allowed to grow as a complete tissue. We now have many other tissues that we can work in, such as the mini brains and hearts and vessels and things like that. So this area is progressing very rapidly. Now, I'd like to just talk to you about two different models of modeling human disease. One is on the left is the type which is the most commonly used in the world, and that's essentially to collect fibroblasts or cells from different patients, and then to have the different patients, such as 1 through 6, 6 being in the red here, where you can make diseased iPS cells, and then you can compare those and to see in diverse populations how those cells behave. You can even correct those different iPS cells, and that has been informative. However, that because they come from diverse populations, the mutant lines, even if they're, say, variants of mutants in the exact same gene, can't be compared directly to each other because they have different genetic backgrounds. In contrast, one can make an isogenic series on the right, as illustrated on the right, with this WTC listed. And WTC is the wild type C line that we actually have provided to the community, and is actually now becoming sort of a common platform for studying human disease. But one can do this with any wild type iPS cell. And so if you take a wild type cell line like this and now make an allelic series where you now see multiple different versions of the same iPS cell, you can make an allelic series of diseased iPS cells, for instance, make disease mutations. And what's different here is that now you can take, say, for instance, C1 and compare it directly with C6. And you can say that are they really different from each other in a molecular manner with the background completely controlled. And so one can actually now tell the difference between subtle differences between, say, mutations in the same gene or the same type of disease. This actually making an isogenic series is actually faster, less expensive, and easier to reveal the mechanisms of genetic interactions. And you can utilize common resources such as the Allen Institute for Cell Sciences, as indicated here, which distributes many different versions of this cell line. So when I say it's faster, it's actually about 1/5 of the cost of doing the same experiment using patient lines. And that's because, especially if you want to make a cell type, the disease cell type, like cardiac myocytes, like neurons, that every cell line will behave differently and have slightly different phenotypes just because of the genetic background. Whereas if you start off with a well-characterized iPS line, it's much easier to make that end tissue. And that's actually why it's so much easier to do. And so the wild type C in particular, but in many other kind of well-behaved cell lines, you can have even high school students essentially growing these cells and really doing this routinely, and makes everything a lot easier as a result. So this common platform for human biology as wild type C, as I indicated here before, the reason that it's so useful is that it has a stable karyotype and has stable pluripotency. There's over 100 mutant variants in my lab and then the Allen Institute and many others. And there's many tool lines such as TET-induced CRISPR, CRISPR-I, and the genome is fully sequenced and is available to academia and industry. The line is also available publicly at the Coriell and the Y-cell cell banks and many academic cores as well. Now, one of the great advantages of this line, particularly with the Allen Institute, has been that they have now actually labeled various different structures, and they're going to be labeling over 100 different structures in the cells. And so this is a picture here of lamin B tagged wild type C cells. And this is a complete lawn of cells that is a cluster of cells, but only the nuclei are labeled. And this is very, very powerful because, in fact, that these cells then, no matter what they do, you can, in terms of differentiation, you can always follow this lamin B staining. And they've marked many other structures, for instance, structures in the cardiac cells, structures in many other areas interest to cell biologists. And this is an incredibly useful resource. Now I'm going to switch now to the second topic, which is how we actually think about not just using iPS cells to decode disease, but how we think about using them as a model for genome surgery to cure rare disease. And all of this work is really in close collaboration with Jennifer Doudna and her institute, the Innovative Genomics Institute. So why do I call it genome surgery in particular? And so I just would like to talk a little bit about that name because there's sort of many meanings to that, and one can think about. So the problem with approaching CRISPR disease with CRISPR is that most diseases require many different approaches. So there's in this bar graph, this pie chart here, we see actually that represents 7,000 diseases, and relatively few of these 7,000 diseases are common mutations. So you can think about a gene drug, one drug that would actually be given a gene vector that would be used in many, many people. Now, those are obviously of great importance. There's no question about that. And the pharmaceutical companies and biotech companies are pursuing those. But the vast majority of genetic disease is really not amenable to that sort of standard model. And one thinks about a different model, perhaps, to approach these rare diseases. And one can think about the surgical model, where every patient is different. And it's useful to think about the surgical model because there's different regulations, different funding, different training that one would actually use in a surgical model. And so I just remind you that there's really different types of heritage and history for medicine versus surgery. And these different historical origins are in the actual word physicians and surgeons. The Columbia, the School of Medicine, for instance, is physicians and surgeons, and they come from very different backgrounds. Physicians, of course, come from the tradition of prescribing different types of medicines for people, whereas surgeons have always been associated with sharp knives and being able to cut things out. But the difference goes beyond that in the sense that that traditionally medicine thinks about using drugs to cure a disease, and billion-dollar drugs, which are obviously also very important, where surgery takes on each person as a rare disease and takes a different surgical approach. And so, for instance, when people did the first heart transplants, they were just happy that the patient survived, but they weren't actually thinking about how to monetize the 10th heart transplant. They were actually thinking about the next way that they could do it better. And each time in an iterative process, these processes got better. And perhaps I'm postulating that genome surgery for these rare diseases will add up together so that eventually that we will be able to approach genetic disease somewhat like a surgeon, where different patients have different remedies. And so what would we think about genome surgery as well in terms of anatomical terms? On the right, you can think about restoration, and particularly you think about a single base pair change from an A to a G, or insertion, where you actually could insert a piece of DNA as shown in the center, or one could think about excision, where, say, there's a disease part as represented by the red area, and now that actually can be potentially removed. And it turns out that in non-dividing cells, that this is by far the easiest to do with CRISPR. And so we actually focus on excision as a therapeutic mode because it's just stunning how efficient this can be. In certain cell types, we see over 80% of the cells where we have a precise excision just at the right place. And this is not just for what we're doing, but actually, if you think about where therapeutic genome surgery has already gone on, it is used as excision. And so, for instance, the CCR5 knockout is essentially an excision of part of the gene. The de-repression of fetal hemoglobin for sickle cell and thalassemia is really the most commonly pursued method of a therapeutic editing that is being used by multiple, I think now over 5 biotech companies. And then the exon skipping, of course, with Duchenne's muscular dystrophy, also uses excision to really have its benefit. So where we are right now in terms of how we think about the genome surgery process I'll tell you about is sort of how we think about choosing the disease, as shown as the arrow on the left. And really in about 2013, we began that process. And then we actually thought about many different diseases, but then we chose a specific diseases that I'll tell you about. And then you have to find the patients, make iPS cells, and then make tissues. Which is our current focus, and test the edits. And I'll be telling you about getting to that second arrow and some of our beginning test edits. So for proof of concept, we thought about multiple different types of therapy and tissues to work in. The heart on the left is actually what we'd worked on before, but we're focusing most of our effort on the retina and motor neurons, as shown on the right. And the reason that we are is because there's several advantages, at least for proof of concept studies. Certainly the heart is a very important organ, and the diseases there are one that we would like to address. However, there are 5 billion cells in the heart, and the dominant, and also the types of disease, the genetics of the disease are not as well understood as in the other organs. And then the risk of potentially having an adverse effect if one were to treat the heart is much more severe. So for proof of concept studies, it's less than ideal. In contrast, the retina and the motor neuron, you can actually probably edit 100,000 cells and see a measurable effect. I mean, you probably want to edit a lot more cells to cure a disease, but to have a measurable effect, because you can measure effects in these cells, is very important. The other thing is that you can have the dominant negatives, as I'll indicate later, are a type of genetic mutation, which is very well characterized in retina and motor neurons. And the risk, although significant, is much less than the heart. So we're focusing on these tissues primarily so that we can move the field forward and do these types of proof of concept studies. In addition, we are looking for not just a particular organ, but within that organ, we have to think about how, which would be the disease course that you would ultimately want if you were to design a trial. And so, for instance, in the bar on the box on the left, we can see that there are, in fact, this is a disease course which starts early, so you can identify the patients, and then as they progress in years, the patients continue to get worse. So there is a progression of the disease, but they don't get worse suddenly. And so having this predictable type of decrease is something which is advantageous, so that one can, in fact, perhaps intervene. And by making an intervention, as indicated by this white arrow, that if at any time, if we can make a change, that we could then see an effect, and we would know that we've done something positive. Whereas this is in contrast to the middle time course, where we actually see that the disease waxes and wanes. And with the waxing and waning, it's very difficult to know at any time if our intervention is what caused the change, or whether this is just the natural disease course. And so, although these other diseases, such as for instance, this kind of represents an ALS course, these diseases are incredibly important, but at the same time, they're very difficult to do proof of concept studies. Whenever we can, also our tool of choice is the Cas9 RNP, which is the RNA Cas9 protein complex, or RNP. This is a DNA-free genome scalpel, and this is a very powerful, particularly in the eye, where this protein complex can be readily taken up. The advantage of this is it causes the least possible risk to the patient, because the Cas9 is a protein which actually degrades within less than a day in most cases, and and is complex to a guide RNA, which is also synthetic. This reduces the cost of the material which goes into the patient, and also it reduces the risk, because there's no DNA involved. There's no DNA that can be integrated into the patient. This is particularly important because the Cas9 is a bacterial product, and as a bacterial product, is known to cause immune responses in humans. And so, therefore, you don't really want the bacterial product to be around for a long period of time. Cas9 RNP is rapidly degraded, and therefore, we can immune suppress for a short period of time, but you don't have to worry about a long period of time. Now, the disease that we're focused on the most in the eye is a disease called BEST1. BEST1 is a model system and is a disease which has proven dominant negative mutations causing blindness and allele-specific activation. It's called BEST1, actually, because Dr. Best described it over 100 years ago. But it's also, we consider it to be very aptly named BEST1, because it really is the best for modeling this type of genome surgery. Its target tissue is RPE that can be grown from a patient's iPS cells, and the RPE preferentially takes up Cas9 RNP. There's a gradual progression of the disease, and the treatment outcomes can be measured. Bestrophin and Best disease occurs in the RPE, which are shown as the blue cells. And the blue cells, as indicated below, have embedded in them the photoreceptors. The photoreceptors slough off every 10 days and is eaten by and phagocytosed by the RPE. And so this RPE is a professional phagocytosing machine that takes up protein lipid complex. And essentially, that's what we're going to be feeding the cells when we give them Cas9 RNP, is a protein lipid complex. And so targeting is really driven by the fact that the cells can be that preferentially take up this complex. Now, bestrophin is a chloride channel which sits on the surface membrane of the cell, causing somehow contributing to the phagocytosing process. And when the bestrophin is diseased, then you get a lack of phagocytosis, and that's what seems to be correlated with the disease. So the way we think about dominant negative alleles is that we have, this is indicated here with the structure of the best protein, BEST1 protein. You can see here that it's a homopentamer that is made by the BEST1 protein. and you have 50% of it coming from either the healthy or the dominant negative protein. And in this case, you can see that in some cases only one subunit is taken up by the best one dominant negative allele, or sometimes all of it. But in any case, these channels are now inactive. And so even though only 50% of the protein is disease causing, because it's this homopentamer, 97% of the pentamers are poisoned in one way or the other. So essentially, the phenotype of this disease, of the severe types of mutants, are in fact very similar to a patient with a knockout of the best protein. So we have a mechanism that we think is there, and obviously, what we would like to do then is to be able to remove and eliminate the dominant negative allele to potentially cure the disease. And if we remove the dominant negative allele, as shown by these red X's, then only the healthy protein would be made, and we could theoretically cure the disease. But how do we know that this would work, that only one copy of the gene would actually be sufficient for normal visual function? Well, fortunately, human genetics, the excellent human genetics in the eye, actually gives us the answer. There are actually humans, maybe some of the people that are listening to this presentation now, actually only have one copy of best one, which is functioning. And that one copy is known to be completely healthy and have completely normal vision. And how do we know that they have normal vision? Is that some unfortunate people who have only one useful copy of best one then have children with another person who has only one copy, and they have children with, unfortunately, no bestrophin. And those children have severe versions of best disease. But now we know for sure when we go and examine the parents that those parents have absolutely normal vision, and therefore essentially the experiment has already been done for us. So if we can, with CRISPR, now go in and take out just the diseased version of the gene with CRISPR, we should have a healthy subjects. The other disease that we're working on is the Charcot-Marie-Tooth disease, or CMT, as a model system. It has proven dominant negative mutations, a crippling disease, and we can use this to test allele-specific editing, particularly in motor neurons. Motor neurons can be grown from patients and iPS cells, and I'll show you some pictures of those. And you can deliver the Cas9 RNP to motor neurons, but unfortunately, the Cas9 RNP is going to be a bit challenging. So we may have to use viruses and take advantage of the tremendous research that other people have done to make viruses that can actually target the motor neurons. There's gradual progression of the disease, and the treatment outcomes can be measured in the sense that you can actually directly target, when you target someone's motor neurons, and if you make those motor neurons better, we know exactly what motor neurons do, and we can test that by motor function. So the Charcot-Marie-Tooth 2 is the type that we're most interested in right now. And that's because they have clear dominant negative genetics. And the dominant negative genetics are exactly the same as the dominant negative genetics that we were showing, or similar to the ones before. In other words, that the mutations in one allele causes a disease, and that a complete loss of that allele is completely compatible with normal physiologic function. So the genes that we're working on here are shown as mitofusin 2, neurofilament light chain polypeptide, heat shock protein 1b, and the tRNA synthesases, which are each can result in a type of motor neuron disease, a loss of motor neurons sometimes, so that people end up requiring a wheelchair. So the basic idea is that we would like to be able to sequence, this is for the best one or for the other types of, or the Charcot-Marie-Tooth, that you'd like to actually take the person who has the disease, to isolate to sequence them, and everybody will be sequenced essentially in fairly soon, that we'll be able to, almost all the patients will be sequenced. And and then you'd be able to say, okay, so I see where your mutation is that causes disease, as indicated by this this kind of fireball here. And I could say that if that mutation, and we might not have a Cas9 that targets that particular spot, and a lot of these dominant negatives have many different spots that are causing the disease, and many different mutations that cause the disease. But we could say, you know what? But your mutation is in cis. In other words, it is on the same chromosome as, say, guide number B and guide number E, and we already have that. And these types of polymorphisms are actually very common in the genome. So we're targeting the Cas9 to those specific common alleles. And then the idea would be that we could then take B and E and put them together, and then to delete the disease allele, have the normal allele intact. The advantage of using the common variants for excision is that we won't have to make guides for all the different rare mutations that often occur, dominant negative alleles. But we potentially could, in fact, have a panel of pre-tested RMPs or guides, which actually allows us to delete the allele. Now, this goes for, as I said, best one as well as the Charcot-Marie-Tooth. Now, of course, we don't want to test this in people without testing it in their cells first. And one of the great things about the motor neurons, as well as the RPE, I didn't have time to show you the RPE, but we have beautiful RPE growing. And we have beautiful motor neurons also growing from the iPS cells. And as indicated here, we can actually take the iPS cells and in a few days actually make motor neurons. And the way that we make motor neurons from iPS cells is to force express transcription factors. So they're indicated here on this diagram of a vector. We have 3 transcription factors shown in green, red, and purple, and each of these transcription factors contribute to making motor neurons. When they're overexpressed together using the TET system, we can get motor neurons in just a week, as indicated here. All the purple cells indicated here are expressing a unique motor neuron specific protein. And we can do this in 96-well plate format or other types of plate formats. And because the system has been stably induced into motor neurons, we can get them to grow essentially on demand. And this greatly improves our ability now to model the system. Now we have these cells are from a control cell type, wild type C, but we have similar results now with other types of cells from patients, for instance. And indicated here, we have also really robust changes that are most obvious when the cells are in spheres. So on the left is in a 96-well plate, and shown here is a sphere of about 2,000 cells, which are expressing, now you can see the arborization coming out of them. To take a closer look at that arborization, you can say here on the day 1, 2, 4, and 5, you can see that after the induction of doxycycline, a rapid induction of these axons. And we're very excited about this. We think that this will allow us to grow tissue, which we can then test. And we're right now in the process of taking the cells that have CMP and comparing them with and without editing. So the steps in genome surgery are, just in summary, are that our currently focus is making the tissue and sort of starting the edits, and we have very robust allele-specific edits, and we're sort of in the midst of testing now hundreds of those on the cell types that we have. The major challenges, of course, are quite significant. Can we deliver Cas9 RNP to the target tissue? Can we utilize cell-specific surface markers? Will we have to use viruses? Can we control maybe Cas9 activation within specific cells? Controlling DNA repair for small excisions or even large excisions are still largely unexplored. This is an area of great research, and we're now tackling that and using these clinical samples as a way to start challenging our very knowledge of how DNA repair goes and happens in these differentiated tissues. This is studied in cancer for sure, cancer cell lines, but not in these differentiated tissues, and that's critical for our experiment to work. Obviously, we want to move towards clinical trials, and clinical trials will require us to have, bring together a number of people, including clinicians, regulatory affairs people, animal trials, and so on. And this process is challenging, but also exhilarating because we really are making progress on this front. Do we see genome surgery as a new standard of care? The long-term goal is to make genome surgery standard of care, in fact, as a sustainable model. And the surgical model, we believe, is really a way to use this in a broad range of types of diseases, using, of course, these very relatively rare types of disease. But I think that will provide the foundation for something larger. We can also reduce the cost, hopefully, to save money for society while reducing suffering. The expanded types of edits in the clinic can only be done really with these sort of test cases. So for instance, allele-specific editing, DNA excision, even base editing or targeted methylation are all things which are things that we would like to use for genome surgery in the future, but would be difficult to test in a larger group until we've had these certain test cases. And some of the test cases that I described today have a very strong, compelling reason for that. The mechanisms of disease are also incredibly important because we can actually really, by doing these edits, we're actually testing the mechanism, and we can test to see whether or not this can actually cause, we don't really know the mechanism for most disease. And just by doing the editing, we actually are proving that point for a particular case, saying this gene is causative, in fact, and it can, in fact, really potentially be reversed. So in summary, I think that we have been careful. We're really at the beginning of this adventure. I don't think that this by no means a sort of finished story. We have careful choice of the disease targets. We have two diseases general types of disease that we're working on, BEST1, and then several genes within CMT. And then I talked a bit about genome surgery and future directions. And so with that, I at the end of my time and I'm ready to take questions.
Thank you, Dr. Conklin, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, what do you think genome surgery will look like 10 years from now?
Well, I think it's a very good question. I think that it will definitely be quite different in the sense that certainly there will be clinical trials that have proceeded both for relatively common diseases such as sickle cell and thalassemia. And so we'll know for a proof of concept that whether or not this type of editing can be used to treat disease. And this will be important because these are incurable diseases that will have potentially a permanent sort of solution for. Furthermore, those are the ones that are in the commercial realm. In the non-commercial realm, in the academic realm, will be taking on a much wider variety of diseases. And there'll be clinical trials specifically in the retina and nervous system for a wide variety of diseases. And this is going to be incredibly helpful and for a number of patients, because these are also diseases that are without any cure, and they have at this point there's really no hope of finding a medicine. But now with this new therapy, that we could treat a much larger group of people. So even though each of these diseases are very rare, the fact that as a sum, there's many more of them, and that will have a bigger impact on the overall population. I think that we'll be moving from the proof of concept stages, where we're trying to actually make focusing on diseases which have, like I say, the lowest risk and the highest impact, to areas where we can actually go to higher risk situations, such as in the cardiac system. We can be going into perhaps competing with other types of therapies, for instance, with transplants, for instance. So in heart transplants or kidney transplant or kidney or liver transplants, right now, most of the time, people would choose to have a transplant if they need such a therapy. But as the technology becomes safer, I imagine that it will be used in those settings as well. And then a wider variety of diseases can be treated there as well. So I think, yeah, so I think it's going to become moving towards sort of a standard of care in certain diseases, and then as a research tool in a much wider group of diseases in the 10-year horizon. And gradually, those experimental therapies will then be turning into standard of care.
And our next question, do you think genome surgery will be too expensive?
Well, I think that too expensive is a difficult sort of thing to define. But I do think that, and certainly a concern for people, is that that this is a therapy which would be really unattainable by many people. But in fact, there's really nothing that expensive about the technology. So, for instance, a Cas9 RNP in particular, the cost of goods for going into somebody without the medical costs, I mean, the physician costs or anything like that, is actually quite low. We're just talking about a recombinant protein and a and the synthetic RNA, and then with some lipid and around it. So there's really no reason that it needs to be an expensive therapy. Of course, the research and development will be potentially expensive, but that's something that we can hopefully we can try to cover with philanthropy or other purposes. So it's possible that at a genome surgery itself, for certain settings, actually could be reasonably priced. And in fact, especially when you think about the cost of blindness, cost of having someone be crippled and so on, that it'll be much less than those costs and burdens on society. So I think that when compared, say, for instance, to a heart transplant or other types of major surgical procedures, this actually could be on the lower end of the costs as we move forward.
I would like to once again thank Dr. Conklin for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.